When it comes to health issues, over the past 2 years, COVID has undoubtedly taken the stage. However, there's been a condition humanity has been fighting for at least half a century. In fact, people with this condition are more at risk of developing severe symptoms from COVID. Russia, in particular, has one of the highest mortality rates in the world when it comes to obesity. In 2017, 15.96% of deaths in Russia were attributed to this condition, and the numbers are only growing each year. Overweightedness or obesity was found in more than 60% of adults surveyed in Russia, approaching prevalence levels in the USA and higher than the prevalence reported in any recent survey in the European Union. We'd just like to reiterate that we're not talking about variability in healthy levels of fat, which have previously been stigmatized by our society, especially in women who naturally have higher fat levels than men. People questioning old beauty standards is perfectly valid and even justified. However, this aspect is a whole separate discussion which has little to do with the physiological aspect that we're interested in as biologists. Although we believe that obese people obviously shouldn't be isolated socially or blamed for their weight and appearance, it's crucial to remember at what cost obese people are living with this condition. Unfortunately, obesity is dangerous, and the most serious consequences of obesity on health are diabetes, myocardial infarction, and major cardiovascular events. In particular, diabetes, as a consequence of caloric excess, shows a direct association with other comorbidities, such as hypertension, which is positively correlated due to vessel damage. Our project represents an attempt at therapeutically managing obesity and other conditions. Well, how do we fit into all of this, and who even are we? Please allow us to introduce ourselves. We're a team from the Russian Federation, and the very first iGEM is from Siberia, a vast territory on the east of Russia. This is why we chose the name Siberia. Let's find out a little bit more about each of our team members.
Hi, my name is Daniil and I am a team leader. I've been working on team management, graphic design and social media this year. I hope this experience will be a foundation for my next projects in the biomedical field.
Hi, my name is Marina. I'm a team leader. I've been designing experiments and doing some of them by myself. Participation in iGEM taught me a lot, and I can say with confidence that my scientific career is just beginning.
Hey, I'm Artemi and I'm the team leader. iGEM for me is a tryout of my first scientific life and an immeasurable source of experience.
Hi, I'm Uliana, a member of the iGEM team responsible for lab work and media. iGEM gave me opportunity to be a scientist on my own and try out my future profession in business.
Hi, my name is Daria and I'm a member of the media team. I was responsible for organising collaborations with other teams, but I also did research into an in vitro model for our project. For me, iGEM is an approximation of a real scientific process, and thus it's a sort of preparation for my future scientific career.
Hi, my name is Anna and I am a member of media team. I've been working on illustration and design. And for me, iGEM is a challenge in which science and creativity tightly intersect.
Hi, my name is Alexander and I'm a member of the Dry Lab team. For me, iGEM is an opportunity to participate in a very interesting and important project, as well as to practice my bioinformatics skills.
Hi, my name is Evgeny and I've been working in a Dry Lab. For me, iGEM is a test of my abilities, and I'm glad I didn't miss an opportunity to participate in this event.
Hello, my name is Anastasia. I am involved in the educational part of the project. I joined the team because I thrilled the idea of bioengineering.
For several months, we have been working together and developing our project, facing and overcoming various challenges. And now, we're finally ready to present our project Leap to Brain.
Leap to Brain is a novel delivery system for the treatment of obesity. The idea is to use symbiotic bacteria living in the nose as a delivery platform. The modified bacterium secretes a chimeric LEAP2 protein which crosses the nasal mucosal barrier and travels to the hypothalamic region of the brain via the trigeminal nerve. LEAP2 is an endogenous antagonist and inverse agonist of the GHSR1 receptor found in the hypothalamus. This is a receptor for the hormone ghrelin. Ghrelin's hallmark functions are its stimulatory effects on food intake, fat deposition, and growth hormone release. It has already been shown that an increase in the ratio of LEAP2 to ghrelin corresponds to weight loss in mice. Supposedly, this happens due to an onset of a feeling of satiety, which leads then to a decrease in food intake. The results of preliminary study on this effect suggest that LEAP2 may be used as a treatment of classes 1 and 2 of obesity in humans. However, a separate study on mice showed that systemic administration of LEAP2 had no effect on high-fat diet intake, whereas centrally injected LEAP2 reduced binge-like high-fat diet intake. This finding points towards a central mode of action of LEAP2, indicating the need for centrally acting formulations of the compound, for example by intranasal application. We believe LEAP2 is a perfect candidate for nose-to-brain delivery. It has an average size of a typical protein, 65 amino acids, so the results of its transport can be used for other similar human proteins. Moreover, the small size of the protein is expected to facilitate secretion into the culture medium during our test experiments. According to various sources, first 10 to 14 N-terminus amino acids are sufficient for binding to the GHSR1A receptor, so the other end of the protein is open to different modifications. To detect our protein in the medium and eventually in the brain, we use MIGTAC, conjugated to LEAP2 at C-terminus. It will allow us to visualize the results of both the secretion into the culture medium and theoretically in vivo delivery to the brain. For the increase in cell permeability level, we combine our chimeric LEAP2 with the cell-penetrating protein LMWP, which is bound to MIGTAC, leaving it in the middle of our protein structure. Although we did not have plans on demonstrating the physiological effect of the protein, we carried out in silico modeling of the putative structure of the fusion protein to ensure that the MIGTAC and the N-terminus are open, both for the production of the plasmid due to the reduced activity of nucleases and for the secretion of the protein due to the reduced activity of proteases. There is a need for two different bacteria to be used as chassis. We selected for these purposes top 10 and BL21 strains respectively. Regarding BL21 strain, normally when it comes to bacterial secretion of a protein, it is preferable to use the hemolysin A system, type 1, since it provides immediate transport to the extracellular environment. However, in our case, the N-terminus of the protein must remain free, and therefore, the LMWP must be attached to the C-terminus, which means that placing a non-cleavable signal sequence there is no longer possible. Therefore, we decided to try secretion into the periplasm via the SEC and TOD pathways. The former ensures the secretion of unfolded protein, while the latter, on the contrary, allows the transport of the folded protein. Both of these pathways have similar cleavable and terminal secretion signals. This is a somewhat risky decision. There is a great probability that the protein will not be secreted out of the bacteria, so we try to pick several different leader sequences. Additionally, the influence on LMWP on protein secretion is in itself of considerable interest. There is currently no data on how CPPs affect the membrane permeability for a protein from the inside. We were planning to make a control cell line secreting LEAP2 plus MIGTAC without LMWP. Other types of secretion systems are found mostly in pathogenic microorganisms. Some of them have complex secretion signals. In some cases, the secretion signals are not cleaved, like in type 1. Some systems are not well understood, and an attempt of their use is in itself enough reason for a separate project. Building up a regulatory pathway is extremely important for the living therapeutics design, but the choice of nil-connected signals in the nasal cavity is extremely poor. We consider a great result of our brainstorming that we have discovered an alternative. Why do we need an outer signal if we can just help people stick to their diets? We are going to make our bacteria secrete LEAP2 periodically, increasing the appetite when it is dinner or breakfast, and decreasing it after the meal is taken to prevent uncontrolled snacking. After all, circadian rhythms play a crucial role in our health, so why not use them? That's why we've been working on an oscillator for our project. that will allow us to periodically change the level of chimeric protein production and secretion.
It's time to talk about the results of our team's work. Leap to Brain was the first solo project our team has taken part in, and the first real research where we had the opportunity to decide our experimental path on our own. Here's the list of our results of all our decisions. The longer I worked on our project, the less we liked the idea of jumping straight from protein secretion in a culture medium right into testing on mice. A need for some intermediate stage became increasingly obvious for us. Thus, we have developed 2 in vitro models, experiments on which can answer many of our questions regarding the penetration of the mucosal barrier of the nasal epithelium and also the nose-to-brain delivery via the trigeminal nerve. This will allow us to tune our project better and save animal lives. Another great thing about this is that this might also help us answer several fundamental questions regarding nose-to-brain delivery of macromolecules, as the exact mechanisms are currently unknown, and an in vivo model wouldn't be very applicable for this goal. Here you can see the result of colony PCR, which shows colonies with insertion in the correct orientation. This was further verified by sequencing. Here's the result of high-fidelity PCR. The matrix was P-blue script with correct insertions. All bands have the expected length. During our work, we encountered problems with obtaining the expression vector and put a real effort into troubleshooting. Here you can see the pathway which led us to a major cloning issue. The problem was in the purification kit, which had inhibited the restriction. However, it turned out to be not the only trouble, as we have already mentioned. Due to the fact that in the nasal cavity there is little to none satiety-related biomarkers, we've made the choice to regulate our protein secretion by a genetic oscillator. An important parameter in models of genetic oscillators is the period. In our model, we need the bacterium to express the gene LEAP2 approximately 4 times a day, times at which people tend to consume food. Taking into account that a period of 6 hours is extremely long, we choose 2 models to work with. The first model is the repressilator, which is a simpler one, but in which we would have to significantly increase the period. The second is a modification of an existing cyanobacterial circadian rhythm-based oscillator, which is much more complex, and in which we would have to decrease the time period. We plan to continue working in both directions. After that, we plan to engage in the implementation of the resulting model and in vivo testing in order to assess whether the resulting system really meets our requirements.
During our project, we not only did lab work, but we also actively engaged in education and communication with the public. Obesity is clearly an issue here in Russia, so we decided to not only practically develop a therapeutic solution to such a condition, but also to raise awareness about the significance of healthy nutrition amongst young people. While managing our social media, Instagram and conкурсе, we saw a lot of people interested in our project idea. So we decided to carry out a survey that included several questions regarding people's health, eating habits, thoughts on GMOs, and also living therapeutics. This way we've both learnt about our potential audience's demand and analysed the current state of the audience's health. Our survey also helped us make a key decision regarding safety. Our team always considered our project to be more of a fundamental scientific research. It was always more about proving the concept rather than actually implementing it. So originally we only had vague plans of designing a kill switch system. However, our human practices surveys showed that for many people, a method of safely removing or stopping the modified bacteria was a key priority. So we changed our plans accordingly and designed and tested a kill switch system. We'd say the biggest contribution our team has made was in terms of education, and it was the organisation of the August School of Synthetic Biology. This summer school ran from the 21st to the 28th of August and targeted students from regional universities studying biology, biotechnology, biomedicine and other related fields. However, some of our students were only high school graduates. Another key goal of the school was to provide training for educators from all across the country so they could bring synthetic biology to their own classrooms. In the wake of an unfortunate lack of resources on this topic in the Russian language, such an exchange of knowledge and educational materials between specialists is pivotal to raise a new generation of synthetic biologists. This will also help make our work more available to a wider audience and not just the attendees of the summer school in particular. Synthetic biology is actually quite well developed in the capital of Moscow, which is reflected in the proportion of Russian iGEM teams from there. We're currently the only Russian team not from there. In fact, we're the only team from the whole of Siberia, a region the size of 2 Australias and with a population almost the size of that of Canada. We saw our part in correcting this situation and sharing our knowledge, as we have many talented compatriots from all across the country. Our summer school acted as a general introduction to synthetic biology and its methods, and also doubled as a preparation for iGEM and a genome editing section of the local Science and Technology Olympiad. In this way, we aim to not only spread the awareness of the opportunities provided by taking part in international competitions, such as iGEM, but also support local educational events.
We have done quite a job working on our project. Despite certain misfortunes, we are eager to comprehend that problem source and finish the job. We already have plans for future lab work troubleshooting. We also believe that some of our findings and ideas will inspire future iGEM teams and help them design their projects and avoid some mistakes.